Our chapter 9 is about cardiac mechanics as a pump, and that would include cardiac cycle. But before starting with these mechanics, I will give you only a brief overview of cardiac excitation. Since we were talking about the cardiac property of self-excitation, you should know that the pacemaker cells, they trigger the action potential throughout the heart. The heart, it's a multi-chambered organ comprising cardiac muscle. When the muscle contracts, the chamber diameter decreases and blood is forced under pressure from chamber to chamber and then out into the vasculature. The unique design of the heart means that contraction of its various parts must be sequenced for output efficiency. Sequencing is effected using a wave of excitation whose rate of progress is modulated to allow time for blood to move between its chambers. Movement of this wave through time and space creates electrical gradients within the surrounding tissues that can be detected and recorded at the body surface as an electrocardiogram, which is also called ECG. Because the timing and pattern of the excitation, it varies little from one individual to the next, and ECG, it can be used to detect the abnormalities in the excitation pathway or in the overall structure of the heart. Cardiac muscle is, as you know, it is involuntary and striated in appearance. In the heart, there are three types of cardiac muscles. There is atrial muscle, ventricular muscle, and then there is specialized conductive muscle fibers. Cardiac muscle fibers are made up of many individual cells which are connected in series with each other by means of intercalated discs. They are actually the cell membranes that separate the individual cardiac cells. The electrical resistance through these discs, it is very low, which enables the cardiac muscle to act as a syncytium. Now keep in mind that the resting membrane potential of cardiac muscle is that is from minus 85 to minus 95 millivolts, and it is around minus 90 to minus 100 millivolts in the specialized conducting tissue. Cardiac impulse, it rises the membrane potential from its very negative value to a slightly positive value of about plus 20 millivolts. This positive portion is called overshoot potential. Then often the initial spike membrane, it remains depolarized for about 0.2 second in atria and 0.3 second in ventricular muscle. This results in the formation of plateau. The presence of plateau in cardiac muscle, it causes muscle contraction to last 3 to 15 times more as compared to skeletal muscle. Now the velocity of conduction is around 0.3 to 0.5 meters per second in both atrial and the ventricular muscle fibers. And this velocity is 0.02 to 4 meter per second in specialized conductive system of cardiac muscle. If we talk about the refractory period of cardiac muscle, then the atrial absolute refractory period is 0.15 second, while the ventricular absolute refractory period is 0.25 to 0.3 seconds. Moving on to the relative refractory period, the atrium, it has a relative refractory period of 0.03 seconds, while the ventricle, it has a relative refractory period of 0.05 seconds. The duration of contraction of cardiac muscle is mainly a function of duration of action potential. It is about 0.2 second in atria while 0.3 second in ventricles. Now, what is the relation of heart rate and contraction? You see, when heart rate increases, the period of contraction,
Well, so my dear, what I was telling you that the relationship of heart rate and contraction is in such a way that when heart rate increases, the period of contraction, or you call it systole, this period of systole, it decreases. Now look closely at this figure where you can see the action potentials in different types of cardiac muscle. Contraction of the different regions and chambers within the heart is carefully coordinated by a wave of depolarization. This wave of depolarization, it spreads from myocyte to myocyte throughout the myocardium via gap junctions, as you have already learned. A heartbeat is initiated by the heart's pacemaker, and this pacemaker is called the sinoatrial node in the normal conditions, and this node is located in the wall of right atrium. Then atria contract first. So when they contract, they force their contents through the atrioventricular valves, the AV valves, towards the ventricles. Excitation of the ventricles is delayed by the AV node. Now this point is very important. Keep in mind that the ventricular excitation, it is a bit delayed, and it is delayed by the AV node, and then it is coordinated by high speed Purkinje fibers. Conduction velocity through the different regions of heart, it is related to the shape of the action potential. Contractile myocyte and Purkinje fibers, they express fast action potentials which comprise of 5 phases. So there is one type of action potential which is seen in the contractile myocytes and in the Purkinje fibers, and that is a fast action potential type which consists of 5 phases, and these phases are from phase 0 to phase 4. What happens in phase 0? Phase 0 is the upstroke, and this is due to rapidly activating sodium current. Now this sodium current, it then inactivates in phase 1. Now phase 2 is the plateau phase, right? And this phase 2 plateau, that is due to a slow activating calcium ions current. Whereas membrane repolarization, that happens in phase 3. So when does the repolarization happens? This happens in phase 3 and it is affected by a potassium current. The interval between beats, that is termed as phase 4, right? So phase 4 is the resting phase. It forms the interval between the beats. Now keep in mind that the nodal cells, they are slow conducting because they lack the phase 0 sodium current, leaving the calcium ions current to drive the upstroke of the action potential. We are talking about here the nodal cells, the pacemaker cells. That why the pacemaker cells, they do not have a phase 0, because they do not have a sodium current. There is only a calcium ions current which drives the upstroke of the action potential. Inactivation of sodium ions current and calcium ions current during depolarization, it causes the myocytes to become refractory to further stimulation, right? So this is the period of refraction where the cell, it cannot be re-stimulated. It is because of the inactivation of sodium ion current and the inactivation of calcium ion current. Now this inactivation, it ensures against the possibility of tetanus in the cardiac muscle. Nodal cells, which are the pacemaker cells, and why they are pacemaker? Because they express a funny current that is activated during the phase 4, and that causes slow depolarization toward the threshold for spike formation. Now this funny current is regulated by the autonomic nervous system. How? That the sympathetic branch, it enhances this current, so increases the rate of phase 4 depolarization and hence the speed of the heart rate, while the parasympathetic nerve stimulation, it has the opposite effect. The waves of excitation moving through the heart, they generate currents, and these currents, they can be recorded at the body surface to produce an electrocardiogram. As I already told you, this is also called ECG. Now typical ECG recording, that comprises several distinct waveforms which correspond to the excitation of the atria, and this atrial excitation is shown in the form of P wave. Then there is the excitation of the ventricular septum, of apex and the free walls. Now the excitation of this septum, apex and the walls, this is shown by the Q, R and S waves. And then finally there is repolarization of the ventricles, which of course is shown by T wave in the ECG. The timing, magnitude and form of these waves, that can be used to diagnose the defects in the heart function.
Let's now look at the sequence of events in cardiac excitation. The cardiac conduction system is responsible for keeping the cardiac cycle going.
if the cardiac cycle stops for too long, you experience serious consequences. So how the cycle goes on, you see the electrical impulse, it is initiated in the SA node, the sinoatrial node. The right and left atria contract simultaneously, pumping blood into the right and left ventricles respectively. Next event which takes place, that is the impulse, it passes to the AV node, which sends it to the AV bundle, and in the meantime the right and left atria relax. Then the impulse passes into the right and left bundle branches and ultimately into the Purkinje fibers, causing the right ventricle to contract and to pump its deoxygenated blood into the pulmonary arteries and the lungs. Simultaneously, the left ventricle contracts, pumping oxygenated blood into the aorta. Then the ventricles relax while the atria, they begin contracting, and the cycle starts all over again. Heart's electrical signals, as I already told you, they can be measured and recorded digitally to produce an image which is called electrocardiogram. You call it ECG or EKG. And three major waves of electrical signals, they appear on the ECG, each showing a different part of the heartbeat. The first wave, which is called the P wave, it records the electrical activity of the sinoatrial node and the contracting of the right and left atria. The second and largest wave, the QRS wave, it records the electrical activity as the impulse spreads through the right and left ventricles and their resultant contraction. The third wave, the T wave, it records the relaxation of ventricles. So problems with the signal conduction due to disease or abnormalities of the conducting system can occur any place along the heart's conduction system. Abnormally conducting signals resulting in irregular heartbeat, they are called arrhythmias or dysrhythmias. So let me repeat again. So the conduction system, it consists of different parts, and the first part, that is the sinoatrial node. As you can see here, the SA node. This SA node, it's a small mass of specialized cells, and it lies in the walls of right atrium near the opening of the superior vena cava. So what happens that the sinoatrial cells, they generate these regular impulses because they are electrically unstable, and this instability, it leads them to discharge or depolarize regularly, usually between 60 and 80 times a minute. Now this depolarization, that is followed by recovery, and this recovery is called repolarization, but almost immediately their instability, it leads them to discharge again, setting the heart rate. Because the sinoatrial node discharges faster than any other part of the heart, it normally sets the heart rate, and that is why it is called the pacemaker of heart. And firing of the sinoatrial node, it
triggers the atrial contraction.
Well, so my dear, what I was telling you that the relationship of heart rate and contraction is in such a way that when heart rate increases, the period of contraction, or you call it systole, this period of systole, it decreases. Now look closely at this figure where you can see the action potentials in different types of cardiac muscle. Contraction of the different regions and chambers within the heart is carefully coordinated by a wave of depolarization. This wave of depolarization, it spreads from myocyte to myocyte throughout the myocardium via gap junctions, as you have already learned. A heartbeat is initiated by the heart's pacemaker, and this pacemaker is called the sinoatrial node in the normal conditions, and this node is located in the wall of right atrium. Then atria contract first. So when they contract, they force their contents through the atrioventricular valves, the AV valves, towards the ventricles. Excitation of the ventricles is delayed by the AV node. Now this point is very important. Keep in mind that the ventricular excitation, it is a bit delayed, and it is delayed by the AV node, and then it is coordinated by high speed Purkinje fibers. Conduction velocity through the different regions of heart, it is related to the shape of the action potential. Contractile myocyte and Purkinje fibers, they express fast action potentials which comprise of 5 phases. So there is one type of action potential which is seen in the contractile myocytes and in the Purkinje fibers, and that is a fast action potential type which consists of 5 phases, and these phases are from phase 0 to phase 4. What happens in phase 0? Phase 0 is the upstroke, and this is due to rapidly activating sodium current. Now this sodium current, it then inactivates in phase 1. Now phase 2 is the plateau phase, right? And this phase 2 plateau, that is due to a slow activating calcium ions current. Whereas membrane repolarization, that happens in phase 3. So when does the repolarization happens? This happens in phase 3, and it is affected by a potassium current. The interval between beats, that is termed as phase 4, right? So phase 4 is the resting phase. It forms the interval between the beats. Now keep in mind that the nodal cells, they are slow conducting because they lack the phase 0 sodium current, leaving the calcium ions current to drive the upstroke of the action potential. We are talking about here the nodal cells, the pacemaker cells, that why the pacemaker cells, they do not have a phase 0. because they do not have a sodium current. There is only a calcium ions current which drives the upstroke of the action potential. Inactivation of sodium ions current and calcium ions current during depolarization, it causes the myocytes to become refractory to further stimulation, right? So this is the period of refraction where the cell, it cannot be re-stimulated. It is because of the inactivation of sodium ion current and the inactivation of calcium ion current. Now this inactivation, it ensures against the possibility of tetanus in the cardiac muscle. Nodal cells, which are the pacemaker cells, and why they are pacemaker? Because they express a funny current that is activated during the phase 4, and that causes slow depolarization toward the threshold for spike formation. Now this funny current is regulated by the autonomic nervous system. How? That the sympathetic branch, it enhances this current, so increases the rate of phase 4 depolarization and hence the speed of the heart rate. While the parasympathetic nerve stimulation, it has the opposite effect. The waves of excitation moving through the heart, they generate currents, and these currents, they can be recorded at the body surface to produce an electrocardiogram. As I already told you, this is also called ECG. Now typical ECG recording, that comprises several distinct waveforms which correspond to the excitation of the atria, and this atrial excitation is shown in the form of P wave. Then there is the excitation of the ventricular septum, of apex and the free walls. Now the excitation of this septum, apex and the walls, this is shown by the Q, R and S waves. And then finally there is repolarization of the ventricles, which of course is shown by T wave in the ECG. The timing, magnitude and form of these waves, that can be used to diagnose the defects in the heart function. Now my dear students, you should understand that the action potentials in cardiac muscle, they have some very special features. The resting membrane potential of cardiac muscle is about minus 85 to minus 95 millivolts, and the action potential is 105 millivolts. The membranes, they remain depolarized for 0.2 second in the atria and for 0.3 second in the ventricles. Now this is quite a long time. Two very important features of the action potential, especially in the ventricular cardiac muscle, that is its plateau and a special kind of calcium diffusion. Now, if we think about the causes of the plateau, you should know that the slow entry of sodium and calcium ions into the cardiac muscle cells, this is one of the causes of action potential plateau. You must be knowing that the action potential of skeletal muscle that is caused by the entry of sodium through fast sodium channels, which remain open for only a few 10,000th of a second. In cardiac muscle, the fast sodium channels, they also open at the initiation of the action potential, but cardiac muscle has unique slow calcium channels, or you can call them the calcium sodium channels. Calcium and sodium ions, they flow through the slow channels into the cell after the initial spike of the action potential, and they maintain the plateau. Calcium that enters the cell through these channels, it also promotes cardiac muscle contraction. So I hope that I have made my point clear about this cause of the plateau. Another cause of the plateau of the action potential, it is a decrease in the permeability of cardiac muscle cells to the potassium ions. The decrease in cardiac potassium permeability, it also prevents the return of the membrane potential in cardiac muscle. This mechanism, it is not present in the skeletal muscle. When the slow calcium sodium channels close after 0.2 to 0.3 second, the potassium permeability increases rapidly. Potassium ions, thus, they exit the cardiac myocytes and membrane potential returns to its resting level. So I hope that you are understanding the logic of plateau and the causes underlying it. Now we move on to the second special feature, that is the diffusion of calcium for the contraction. You must remember that the diffusion of calcium into the myofibrils, it promotes muscle contraction. The action potential spreads into each cardiac muscle fiber along the transverse tubules, which you call T-tubules. So the action potential that spreads along the T-tubules, causing the longitudinal sarcoplasmic tubules to release the calcium ions into the sarcoplasmic reticulum, right? These calcium ions, they catalyze the chemical reactions that promote the sliding of actin and myosin filaments along one another to cause muscle contraction. Now, this kind of mechanism, that is also present in the skeletal muscle. But another means of calcium entry into the sarcoplasm, however, it is unique to the cardiac muscle. What happens? The T-tubules of the cardiac muscles, they have 25 times as great a volume as those in the skeletal muscle volume. These T-tubules, they contain large amounts of calcium that are released during the action potential. And the story does not end here. You see, in addition, the T-tubules, they open directly into the extracellular fluid in the cardiac muscle. So their calcium content highly depends on the extracellular calcium concentration. So what happens that at the end of the plateau of the action potential, the influx of the calcium ions into the muscle fiber, it abruptly stops and calcium is pumped back into the sarcoplasmic reticulum and T-tubules, and thus the contraction ends. You do it 100,000 times every day, waking or sleeping,
from a moment early in fetal development until the moment you die, the beat goes on. It may speed up slightly when you are working hard physically, or you are excited or stressed, and some people can slow theirs down with meditation techniques. But for most people, it just plugs along, the same thing over and over. What is it? The heartbeat, of course. The beating heart pushes blood around a double circuit, out through your arteries, ultimately into your capillary beds, then across your capillary beds and into your veins, and then back through your veins. The blood passes through the heart to the lungs, and then back to the heart and out through the arteries again. Each complete double circuit takes less than 1 minute. So, if you put your finger on your pulse, you can feel the rhythmic pulsations of blood flow at certain spots around your body. Most commonly on the inside of the wrist or on the carotid artery of the neck. What you feel as you touch these spots is your artery expanding as the blood rushes through it, and then immediately returning to its normal size when the bulge of blood has passed. The entire cardiac cycle, it takes about 0.86 of a second, based on the average of 70 heartbeats per minute. If your cardiac cycle takes less time, your heart is beating too fast, and this is called tachycardia. If there is too much time between your cardiac cycles, your heart is beating too slowly, and that is called bradycardia. The human heart is composed of two pumps. The right heart, which receives blood from the peripheral organs and pumps it through the lungs. And then there is left heart, which receives oxygenated blood from the lungs and pumps it back to the peripheral organs. The heart has a special conduction system that maintains its own rhythmicity and transmits the action potentials throughout the heart muscles. Certain structures of the heart, together called the cardiac conduction system, they specialize in initiating and conducting the electrical impulses that induce your heartbeat, keeping it regular and strong in every part of the organ. And this is the heart's pacemaker. So, how do you define the cardiac cycle? It is defined as the cyclical events occurring in the heart from one heartbeat to the other heartbeat. I repeat again, the events that occur at the beginning of a heartbeat and last until the beginning of the next heartbeat, they are called the cardiac cycle. Each beat of the heart, it begins with a spontaneous action potential that is initiated in the sinus node of the right atrium near the opening of the superior vena cava. The action potential, it travels through both atria and the AV node and bundle into the ventricles. And then keep in mind that a delay of about 0.13 seconds it occur in the AV node and bundle. Why so? It allows the atria to contract before the ventricles contract. Now this figure, it shows the events of the cardiac cycle. You see the ventricles, they fill with blood during diastole and contract during systole. Here there are curves which are showing the aortic pressure, then the left ventricular pressure and left atrial pressure. And then there are curves which you can see. They are the changes in the ventricular volume, the electrocardiogram and the phonocardiogram. You see, phonocardiogram, it's a recording of the heart sounds. Now, my dear students, keep in mind that the spread of the action potential in the heart, it initiates each heartbeat. The electrocardiogram, it is a recording of the voltage generated by the heart from the surface of the body during each heartbeat. Here you can see the P wave. This is caused by the spread of depolarization across the atria, which causes atrial contraction. And atrial pressure, it increases just after the P wave. Then you can see the QRS waves. They appear as a result of ventricular depolarization about 0.1 seconds after the onset of the P wave. And they initiate ventricular contraction, and then the ventricular pressure begins to increase. Here you can also see the ventricular T wave, which is caused by the repolarization of the ventricle. The heart's function is to generate pressure within the arterial compartment. Pressure is required to drive blood flow through the vasculature, delivering oxygen and nutrients to all the cells in the body. The amount of the blood that the heart pumps out on each cycle, that is precisely matched to the metabolic needs. At rest, the needs of the various tissues are modest, and the cardiac output approximates 5 to 6 liters per minute in an average person. Any increase in tissue activity, for example, during digesting a meal or during walking or climbing stairs, then this increase, it requires that cardiac output increase to support the active tissues' increased needs. Raising cardiac output is achieved in part by increasing cycle frequency, means the heart rate increases. But the heart, unlike a conventional pump, it has the unique ability to increase the amount of blood it ejects on each stroke, and to do so with increased force and efficiency. These features, they allow a fit athlete's heart to ramp up the output by as much as 5 folds to 6 folds during strenuous exercise. Matching cardiac output to tissue needs, this is the responsibility of the autonomic nervous system, which regulates beat frequency, the extent of ventricular filling prior to contraction, and the contractile force. Well, let's now move on to the phases of the cardiac cycle. At rest, the healthy adult heart is likely to beat at a rate of 60 to 80 beats per minute. And keep in mind that this is a range, this is not a fixed number. It's not like 70 beats per minute, it is always a range, and it ranges from 60 to 80 beats per minute. During each heartbeat, or you can say during each cardiac cycle, the heart contracts and then relaxes. The contraction, that is called systole, and the relaxation is termed diastole. Now, the phases or the stages of the cardiac cycle, they are, you can see here in the figure. Each cycle, it lasts about 0.8 of a second, and it consists of three main phases: the atrial systole, ventricular systole, and then complete cardiac diastole. So I repeat again that each cardiac cycle, it consists of three phases: the atrial systole, the ventricular systole, and then the complete cardiac diastole. Now, what is atrial systole? That basically is the contraction of the atria. And of course, ventricular systole is the contraction of the ventricles. While in the cardiac diastole, there is complete relaxation of both atria and ventricles. It doesn't matter at which stage of cardiac cycle a description starts, but for the convenience, the period when the atria are filling, this is chosen to describe the cardiac cycle. You see, the superior vena cava and the inferior vena cava, they transport the deoxygenated blood into the right atrium at the same time as the four pulmonary veins, they bring oxygenated blood into the left atrium. So these two events, they are happening simultaneously, that the superior and inferior vena cava, they are bringing the deoxygenated blood into right atrium, while the pulmonary veins, they are bringing the oxygenated blood into the left atrium. So what happens next? The atrioventricular valves, they are open and blood flows passively through to the ventricles. The SA node triggers a wave of contraction that spreads over the myocardium of both atria, emptying the atria and completing ventricular filling. Now here, keep in mind that the atrial systole is of 0.1 second, as you can see in this figure. When the electrical impulse reaches the AV node, it is slowed down, delaying the atrioventricular transmission. This delay means that the mechanical result of the atrial stimulation, means the atrial contraction, lags behind the electrical activity by a fraction of a second. And this allows the atria to finish emptying into the ventricles before the ventricles begin to contract. So this point is very important. This is called the AV delay. After this brief delay, the AV node, it triggers its own electrical impulse, which quickly spreads to the ventricular muscle via the AV bundle, the bundle branches, and also the Purkinje fibers. And this spread, it results in a wave of contraction which sweeps upward from the apex of the heart and across the walls of both ventricles, pumping the blood into the pulmonary artery and the aorta. Now keep note that the ventricular systole, this is of 0.3 seconds. The high pressure which is generated during ventricular contraction, it forces the atrioventricular valves to close, preventing the backflow of blood into the atria. After contraction of ventricles, there is a complete cardiac diastole, which is a period of 0.4 seconds when atria and ventricles are relaxed. So during this time, the myocardium, it recovers, ready for the next heartbeat, and the atria, they refill and they are ready for the next cycle. Dear students, the valves of the heart and of the great vessels, they open and close according to the pressure within the chambers of the heart. You see, the AV valves, the atrioventricular valves, they are open while the ventricular muscle is relaxed during atrial filling and atrial systole. When the ventricles contract, there is a rapid increase in the pressure in these chambers. And when it rises above the atrial pressure, the atrioventricular valves close. When the ventricular pressure rises above that in the pulmonary artery and in the aorta, the pulmonary and the aortic valves, they open and blood flows into these vessels. When the ventricles relax and the pressure within them falls, the reverse process occurs. First, the pulmonary and aortic valves close, then the atrioventricular valves open, and the cycle begins again. And very important thing is that this sequence of opening and closing valves, it ensures that the blood, it flows in only one direction.
Remember, the heart is a double pump, so it has two circuits, heart to lungs and back to heart, and the other is heart to body and back to heart. These are called the pulmonary circulation path and systemic circulation path respectively. Every drop of your blood, it travels around the double circuit about once per minute. So what happens in the pulmonary circulation? That deoxygenated blood, it enters the heart's right atrium from the largest veins in the body, the superior vena cava and inferior vena cava. And when the SA node initiates the cardiac conduction cycle, the right atrium, it contracts, pumping the blood into the right ventricle. When the impulse is passed to the AV node and onto the AV bundle, the right bundle branch and the Purkinje fibers, the right ventricle contracts, pumping the blood into the pulmonary arteries, which take it to the lungs for gas exchange. During the relaxation phase of the atria, the newly oxygenated blood, it flows into the left atrium. The pulmonary arteries, they are the only arteries that carry deoxygenated blood. I hope you already know that. Now, what happens in systemic circulation? That when the SA node, it initiates the cardiac conduction cycle, The left atrium contracts, pumping the oxygenated blood into the right ventricle. When the impulses pass to the AV node and onto the AV bundle, the left bundle branch and the Purkinje fibers, the left ventricle contracts, pumping blood into the aorta. From the aorta, it travels through the arteries and arterioles to the capillary beds and then back to the heart through the veins. During the relaxation phase of the atria, the deoxygenated blood, it flows into the right atrium. So that is the whole sequence during the beating path of heart. The body tissues and fluids, they conduct electricity well. So the electrical activity in the heart, it can be recorded on the skin surface using electrodes which are positioned on the limbs and on the chest, and this recording is called electrocardiogram. This electrocardiogram, it shows the spread of the electrical signal generated by the SA node as it travels through the atria, the AV node and the ventricles. And I have already mentioned that the normal ECG tracing, it show 5 waves which by convention they have been named P, QRS and T waves. So look at this figure closely. That's a very important figure. Here you can see that the heart, it repeatedly contracts and then relaxes and fills. The contraction, that is systole, of course, and the relaxation, that's called diastole. The cardiac cycle, it can be divided into several discrete phases, and it begins with the atrial systole, which pushes the blood forward into the ventricles and completes preloading. Okay, so the atrial systole, it completes preloading. Now after the atrial systole, there is ventricular systole. In other words, the ventricular systole, it follows the atrial systole. Intraventricular pressure rises rapidly and forces the mitral valve closed. Now the contraction is initially isovolumic. Once the luminal pressure, it exceeds the aortic pressure, the aortic valve opens, and as soon as the aortic valve opens, the rapid ejection begins. Aortic pressure, which had been declining during the diastole, it climbs when blood is forced into the arterial system. Now the rapid ejection, it gives way to reduced ejection. The ventricle then begins to relax and luminal pressure falls rapidly. Isovolumic relaxation begins when the aortic valve is forced closed by high aortic pressures. When ventricular pressure drops below the atrial pressure, the mitral valve reopens and there is rapid passive ventricular filling, which is aided by the influx of blood that has dammed up against the mitral valve during diastole.